Hi. We're going to talk about biomolecules, enzymes, and cell transport today. We're reviewing content, so we should already have our foundational knowledge. So that's going to be our vocabulary and the basics. We're going to look to the past, what we've already learned, and look a little to the future to what we will be connecting these concepts with eventually. The teaks that we are going to cover, we're going to cover B5A, relate the functions of biomolecules to the structure and function of a cell. B5C, investigate homeostasis through the cellular transport of molecules. B11A, explain how energy produced during cellular respiration is used for cell transport. B11B, investigate and explain the role of enzymes in cellular processes. And both B3A and B1B, we're going to apply those scientific practices and models to investigate these systems. So those diagrams that we use to help make this more easy to understand, those are those two teaks combined. So that's why we review our teaks, because it gives us the roadmap and what success should eventually look like. We should be able to do and understand these concepts, but also be able to explain them to somebody else eventually. Our big idea here: cells rely on specialized molecules and regulated transport to maintain homeostasis and carry out life processes. What do we need to figure out? Well, we need to look at how molecules and membranes help cells maintain balance and function in extreme environments. So let's think about last unit. You learned that each cell structure has a specific function. Now we're going to explore what those structures are made of and how they get the materials they need. So, again, thinking back, how the cell membrane controls structure. So last unit we talked a lot about structure, and you needed a lot of foundational knowledge in organelles to be successful at that last unit. Well, in this unit, we're going to make some connections here. The cell membrane isn't just a barrier. It doesn't just keep things out or keep things in. It helps maintain the cell's shape and structure. It acts like a flexible wall that holds the cell together. Proteins in the membrane attach to the cytoskeleton inside the cell, giving support and shape. And by controlling water and ion movement, the membrane prevents the cell from shrinking or swelling too much. Remember when we talked about hypertonic and hypotonic? That's where some of these concepts are going to come and interplay with each other. What organelles are used in membrane transport? So we should know all of our organelles, but now let's think only about the ones that have membrane transport involved. Well, the cell membrane, obviously. It acts as a gate controlling what enters and leaves the cell through diffusion, osmosis, and active transport. The nucleus. The nuclear envelope has pores that allow RNA and other molecules to move out to the cytoplasm. And we'll talk a lot about that next unit. Endoplasmic reticulum. Its membrane channels carry proteins and lipids from the nucleus to other parts of the cell. The Golgi apparatus. Its stacked membranes package and ship molecules in vesicles to their final destination. Vesicles, well, these are small membrane bubbles that transport molecules within the cell or out via exocytosis. Mitochondria. I love mitochondria. Its membranes are inside, so its inner membrane helps move molecules during cellular respiration to produce energy, or ATP. So now that we've reviewed that, let's talk about our biomolecules. The structure of biomolecules determines their function in the cell. Remember, structure determines function. The shape and structure of a biomolecule will decide what it can do in the cell. So proteins, well, proteins are made of amino acids that fold into unique shapes. Their shape determines what they can do in the cell. We have a lot of examples because proteins do a lot of things. Well, proteins are responsible for building our enzymes, structural proteins, transport proteins, signaling proteins, and defense proteins. And all of these different proteins have different shapes. With enzymes, all of the different types of enzymes have different shapes. Same with all of these other examples. They're very specific, but they're all built out of amino acids. But these amino acids form very particular shapes. Carbohydrates have ring-shaped sugars that store energy or form strong structures like cellulose. So similar to proteins, carbohydrates are going to do more than just one thing. But they have the same shape idea, but their different shapes will dictate what they can do with that. So those ring-shaped sugars are going to echo the same shape as our strong structures, especially in plant cell walls, that cellulose. Which biomolecule provides energy for cells? So this is getting away from the structure, but thinking more of the function. Well, carbohydrates are the main biomolecule cells use for quick energy. Glucose is a sugar, and it's broken down into during cellular respiration to make ATP, the energy currency of the cell. Lipids, which we haven't talked about, but we will. Lipids are those fats that can also be used for long-term energy storage. But again, this is more function than structure. Lipids, I told you we would talk about them. Lipids are long fatty acid chains and they are water resistant. They're perfect for membranes or energy storage. So, which biomolecule makes membranes? Lipids. Lipids are the main biomolecules that make cell membranes. Phospholipids form a double layer, the phospholipid bilayer, that separates the cell inside of the cell from the outside. Their structure has a hydrophilic water-loving head and a hydrophobic water-fearing tail, which helps control what enters and leaves a cell. In your phospholipid bilayer, you're going to have those tails inside. They're inside, and then the heads are on the outside of the phospholipid bilayer. So each phospholipid has a head and it has a tail, and from there, you're going to get your phospholipid bilayer. Nucleic acids. The sequence of nucleotides in DNA and RNA store instructions for building proteins. These are the DNA is that beautiful double helix form. So we've seen this a lot. We don't really need to stress out about it right now because guess what? We're going to talk about a whole bunch in the next unit, nucleic acids. So some of the vocabulary that you should already be familiar with, but if you're not, here's a nice recap. We have carbohydrate, lipid, protein, nucleic acid, enzyme, monomer, polymer, glucose, phospholipid bilayer, ribosome, and nucleus. Ooh, but these are words that you should already know, either from previous units or from work we've done front-loading in this unit. What have you noticed maybe about some of those words that we've seen before? Well, within this unit, you should already have seen certain words like glucose, fructose. These are both carbohydrates. They're sugars. So ose, that ending, OSE, is going to be a nice indicator to be like, oh, that's a carbohydrate, that's a sugar. Cellulose is also a carbohydrate, it's also a type of sugar, and it has that same ending, that OSE. Ase, so lactase, amylase. We've done a couple labs where we've talked about these specific enzymes. So that enzyme indicator is ASE. Lactase, amylase, fructase, all of these are enzymes, and they're very specific and they work exactly how they're supposed to. When we get to our next unit, we'll see some enzymes like helicase. And so knowing this and being able to anticipate what a word means It's just going to make your life a lot easier as we continue. So we've talked about the four biomolecules: carbohydrates, lipids, proteins, nucleic acids. Well, what do they do? Well, carbohydrates, we know that they have that sugar circle shape to them. They provide quick energy and some structure. For carbohydrate, for that structure, really think about cellulose. That's going to be one of the big structural carbohydrates. Most of the other times when we're talking about carbohydrates, we're going to be talking about that energy. Lipids, well, lipids store long-term energy and make cell membranes. So we already saw how important those cell membranes are. Proteins, well, our friend the ribosome, the organelle, makes proteins, or rather, proteins are made at the ribosome, and they're long chains of amino acids in different formations. And these proteins build structures, act as enzymes, and do cell work. Nucleic acids, these are storing and transmitting genetic information, that DNA and RNA. Some of their functions, because we want to know not just what their structure is, we want to know what their function is, and we've already previewed that. Here's just a nice little abbreviated chart that you could put into a notebook. Carbohydrates provide that quick energy and structural support. Lipids make cell membranes, act as insulation, long-term energy. Proteins, proteins build everything. They're your structure, they act as enzymes, and they do cell work. Nucleic acids, they're storing and transmitting genetic information. So we've talked about enzymes. I mentioned specific ones like amylase previously. Enzyme function. Well, without enzymes, we wouldn't be able to speed up these chemical reactions. Enzymes are really unique. They help our cells be very efficient because they don't get used up. So we don't need to spend a lot of cell energy making them. Instead, we make it and we can keep using it. As soon as we finish using it once, it's ready to be used again. So enzymes, they're proteins, they're made of amino acids. These speed up chemical reactions and they don't get used up. Some vocabulary, and we're front loading the vocab here. We're going to need to know enzyme, substrate, active site, catalyst, reaction rate, denaturation, inhibitor, competitive inhibitor, non-competitive inhibitor, and specificity. So why do we need to know all of this? Well, enzymes are specific. They do not just partner with anybody. So we need to know enzymes, the biological catalyst that speeds up the reaction, the protein, but the enzyme only works with the substrate, and that's the molecule that an enzyme acts on. The active site is the part of the enzyme where the substrate binds to it. So we have talked about this before with the lock and key, and we'll talk about it again. You need to know what a catalyst does, what that word means. Well, a catalyst just is a substance that lowers activation energy for a reaction. So you don't need as much of something to cause that reaction to happen. All of the others are just words that you're going to see so that you can interact with the concepts that you're going to possibly see on test questions or in reading passages. And I love that last word, specificity. Enzymes are gosh darn specific. So we should know which biomolecule builds our enzymes, proteins. Enzymes are a special type of protein, and they're from long chains of amino acids. And the order of the amino acids gives the enzyme its shape, and the shape determines what job it can do in the cell. Remember, enzymes are specific. That lock and key model. Remember, we talked about the enzyme, and within the enzyme, there's an active site, and that active site is where the substrate will bind. Well, all of those things have to be exactly just right for the substrate to bind with that enzyme. And when that happens, the reaction rate is decreased, and that reaction will happen with less being used. And then as soon as the reaction happens, the enzyme is free to be used in the same way again. So the lock and key, the enzyme is your lock, the key is a substrate, and the keyhole where the key fits into, that's your active site. Well, how do enzymes act as catalysts in the cell? Well, they're speeding up chemical reactions in the cell. And because they're a catalyst, they're helping those chemical reactions happen faster with less energy needed. Each enzyme and its specific substrate fit together, and after the reaction, that enzyme that's not used up, it gets reused over and over again. So we'll see a lot of this when we talk about helicase in our next unit. But for right now, think of it like amylase in our saliva. When we're eating something, amylase helps us break things down. And if you've done the cracker lab, so if you've never done the cracker lab, or if you have, I'll remind you. The cracker lab, you eat a cracker like normal, and that's going to be your baseline. And if you eat a cracker, it's kind of dry, it doesn't really have much taste, it's kind of just dry, maybe a little salty. Well, with the amylase cracker lab, you hold a cracker in your mouth without chewing, and you let amylase that's in your saliva, it's an enzyme, you let the amylase really build up and interact with the cracker, but you're not chewing it. And as you do that, the flavor of the cracker goes almost sweet. And that's because amylase changes how the cracker, the carbohydrates in the cracker, are actually going to be digested. It's a pre-digestive situation, but the flavor changes, and a lot of people do not like the flavor crackers change into. It's not my favorite lab to do for myself, but I like to do it at least once a year because it's an easy lab to demonstrate that change. That, oh my goodness. So let's think. Can we explain how enzyme activity or energy loss can affect cell homeostasis? So, this is actually a big thought. Well, let's tear this apart. Let's look at the end of this concept. Cell homeostasis. Well, cell homeostasis is just the cell staying in balance. It doesn't want to have an environment that it's not well meshed with, so that hypertonic, hypotonic. It doesn't want to use energy needlessly. That's why we have enzymes. They're there to help make us use less energy and do it in a very efficient and fast way. And we don't want to lose energy or anything in the process. So let's see how we can think through this a little bit, where we explain how enzyme activity or energy loss can affect cell homeostasis, so that cell balance. Well, enzymes speed up cell reactions, and energy, ATP, powers them. If enzymes stop working or if energy runs out, reactions will slow down. The cell can't stay balanced, homeostasis would be lost. So that's how important these are. One thing that I will bring up a lot in class is lactose intolerance versus lactose sensitivity. So lactose is a carbohydrate, it's a sugar, but we also have lactase. Lactase is the enzyme that helps us digest lactose. And some individuals, they have an allergy where they can't have lactose. Well, that's different. They can't have it, it causes an allergic reaction, sometimes very severe. Other people might have a lactose sensitivity. Well, if you have a lactose sensitivity, your body just might not be producing a lot or a sufficient amount of lactase, the enzyme to digest lactose. So those people might be able to have some cheese, a small amount of ice cream, but too much, and they're going to have very uncomfortable times. Other people might have they might be like, I really want to eat my lactose-based foods, my milk, my dairy, all that. So they'll take a supplement, a lactase-based supplement, a that they got prescribed from a doctor. So at the cellular level, a lot is happening, but sometimes we will expand from the cell to talk about the whole body because it's just easier to make these analogies. So without lactase, you can't digest lactose. And if you can't digest lactose, that's going to cause a lot of tummy problems for people who are sensitive to lactose. And that sensitivity really is just a reflection of their body not producing enough lactase. So hopefully that made the concept a little more easier to interact with. At the end of the day, if you don't have the right energy levels, and if you don't have enough enzymes, your homeostasis, your cell balance isn't going to be there. It's not going to work. So let's circle to cell transport. Cells control what enter and exit to stay balanced. Again, homeostasis. And we front loaded a lot with this with the cell membranes. So how do things move in and out of cells? Well, cells need to take in nutrients and get rid of waste to stay healthy. Cell transport is how materials move in and out of the cell through the cell membrane, the cell's gatekeeper. Some vocabulary, well, we should be aware already of these words: passive transport, diffusion, osmosis, facilitated diffusion, active transport, ATP, pump, endocytosis, exocytosis, concentration gradient, tenacity, and homeostasis. So homeostasis, we've already talked about. It's a cell's ability to maintain stable internal conditions, so it's balance. Endocytosis, I love endo and exocytosis. So endo, enter, enter, endo, the process of taking materials into the cell. So materials are entering the cell. And exocytosis, exit, the process of removing materials from the cell. And then our types of transport. These are, there's a lot of them, but they kind of are very similar to each other. So let's dive in. We have passive versus active transport. Well, passive transport, there's no energy needed. Whereas with active transport, energy is required. You need to use ATP to get stuff in or out. So for passive, the cell doesn't use any energy. There's no ATP that needs to be spent. Molecules move from high concentrations to low concentrations, like sliding downhill. It's easier. Whereas with active transport, the cell uses energy, ATP, to move molecules. The molecules are going to move from low concentration to high concentration, like climbing a hill or climbing up the slide instead of sliding down the slide. The goal of passive transport is to balance concentrations inside and outside of the cell. So the cell likes its environment outside to match the environment inside. You're going to have diffusion, osmosis, and facilitated diffusion as passive transport. Facilitated diffusion is one where some people really get hung up on, but facilitated diffusion, it's like having somebody hold a door open for you. You're not spending energy to open the door, you're allowed to pass right through. That's what facilitated diffusion is at the cellular level. It's going to use those channels, and those channels in the cell membrane are going to act like somebody holding a door open for you, whatever is moving in or out. Active transport. The cell can move materials even when it's against the natural flow, but they have to spend energy to do so. Protein pumps and endocytosis and exocytosis. So entering the cell, exiting the cell, protein pumps, those are going to be your active transports. And it's because the cell has to spend energy to do that. So just a quick little chart if you needed to add this to your notebook for whatever reason. Maybe you missed that day and you don't already have this. So passive transport, there's no ATP. It's high to low. Diffusion, osmosis, facilitated diffusion. It's to balance or equal the environments. This is like rolling downhill. It's very easy. There's no energy used. It just once it's in place, it's going to happen. Whereas active transport, it requires that ATP. It's low to high. There's protein pumps involved, endocytosis, exocytosis. It needs to gather or remove specific materials. And it's that climbing uphill. It's harder. It costs energy to do. Ooh, this is a great question. What happens to homeostasis if diffusion is blocked? Well, diffusion gets things in and out of our cells. And think about all the reasons why we would need to bring things in or push things out of our cells. If we can't, what is that going to do to the cell balance? So you should already be forming some really good responses mentally. Well, diffusion is how cells exchange materials. Diffusion allows oxygen, carbon dioxide, water, and nutrients to move across the membranes or within the cell without using energy. It's essential for keeping concentrations balanced inside and outside of the cell. That's why we have these different diffusions, our active and passive. If diffusion stops, oxygen can't enter, the cells can't make enough ATP through respiration. And remember, respiration gives us far more ATP than our anaerobic. So we like oxygen being present in the cell. It helps us make better energy conversions. Carbon dioxide can't leave, so you're going to have a toxic buildup. You're going to change the pH levels. What happens to our enzymes if we change the pH levels? Well, enzymes can denature, they can turn off if we change the pH too much. Water and solutes become unbalanced. Cells may swell or shrink, hyper or hypotonicity, and nutrients and waste can't move. So your metabolism is going to slow or stop. Can we predict how energy use connects to cell balance? So, if we think about this, what's the cell balance? Well, that's homeostasis, that maintaining what is going on within the cell. And then we have our energy use, ATP. So, how is ATP being used, and how can we connect this to our homeostasis? Well, energy use and ATP, they go hand in hand. Cells use ATP as their main energy currency. Every process in the cell, from active transport to protein synthesis, requires ATP. When energy demand increases, cellular respiration and mitochondria speeds up to make more ATP. Cells must balance energy intake and output to maintain stable internal conditions. Too much energy without replenishment would cause energy deficit and cellular stress. Excess energy intake can lead to storage molecules. That's going to be your glycogen or fat. So if you take in too much, you can start building up too much of your glycogen or fat stores. Because remember, lipids are long-term energy storage. So if you take in too much, but you're not using it, that's going to be that storage molecules coming into play. Enzymes that control metabolism depend on energy levels, our ATP and ADP ratio. When energy is low, enzymes activate pathways to produce more, so that catabolic. And when energy is high, enzymes activate pathways to build molecules, anabolic. So your enzymes are trying to be efficient and use what you have at the right times. It's time sensitive. There's some time delays. We also have feedback loops. Well, cells use feedback mechanisms to regulate energy. We have negative feedback, which is going to prevent overproduction, and positive feedback can amplify responses in emergency. That's going to be that fight or flight. If you get really scared, somebody's coming at you with a biology test that you were unprepared for, you're going to feel it in your body, not just in your brain where like, oh no, I didn't study. You're going to feel it in your body. Your heart rate will increase. You might start to sweat a little bit. You're going to get maybe flushed on your cheeks. The blood is going to get pumping because your body's like, oh my goodness, we are stressed out. Maybe we have to run away. So you have different feedback loops, and we haven't talked at length about them, but they will keep coming up over and over again. So let's remember the cell membrane controlled structure. And now we're seeing how it controls that balance. Why? Why is any of this important? Well, every cell process from energy use to growth depends on biomolecules working together to keep life in balance. So let's practice a couple star style questions. Which biomolecule provides the main source of energy for cells? Our answer options are lipid, protein, carbohydrate. Okay, hopefully you have an answer. Carbohydrate, amazing, woo! Those are those sugars, that glucose, fructose, galactose. So biomolecule that provides the main source of energy, carbohydrate. Lipids provide that long-term energy, not the main source of energy. All right, another question. Which statement best describes the role of enzymes? So A, enzymes are changed during reactions. B, enzymes speed up reactions without being used up. All right, we've talked a lot about enzymes. Hopefully you have the correct answer. Enzymes speed up reactions without being used up. So hopefully that's what you got. If not, make note of it now. All right. What would most likely happen if a cell ran out of ATP? A, it could still perform active transport. B, active transport would stop. So what would most likely happen if a cell ran out of ATP? Active transport would stop. Active transport requires ATP. If we don't have it, we're not going to be able to do it. So let's look a little bit ahead to what we're going to be talking about. You now know how enzymes work and how materials move to keep cells stable. Next, we'll explore how cells grow, divide, and specialize, processes that rely on these same molecules and enzymes. So this bridge to the cell cycle differentiation and regulation. The enzymes that help with transport and reactions now are going to be something that we talk about in terms of helping control the cell cycle. So I can't wait to talk more about this with all of y'all later.